I'm going to be talking about explorations of telomere biology and in the context of human aging. So let me just tell you the plan, what I'm going to talk about. I'll take you through a very brief overview without a lot of specifics about just the basics of telomere basic science and how that has been able to inform the impacts of when that loss of telomere maintenance, which is critical for cells, when that loss happens, what happens in actual humans. I'm going to present a very high-level view of this because I think we can see a pretty coherent picture that has emerged through a lot of labs studies. I won't be able to give credit to all of the wonderful groups, both in basic science and in more translational kinds of science and human genetics, who've really contributed to what I'm going to summarize all too briefly. Because what I wanted to do was to take you into something that, being Professor Emerita, I could choose my time, and because of COVID, I couldn't get on millions of planes every week, and so I stayed at home and I thought, I'll look at the public databases and try to look at telomere maintenance based on all of the fundamental biology that we know and the disease impacts of losing telomere maintenance on things that we can look at in terms of cells, directly experimentally, and when I say we, I mean the field. I'm just looking at data sets and effects of things that are very vital to cells, nutrition. I'll talk about examples of each of these. I'll take you through this. Stress, toxins even, mechanotransduction, implications for space, microgravity even. And what I'm going to do is show you how I've been looking at the data in a kind of a different way, and I'll walk you through that. But it's all data that's available. Primarily, I use the NCBI genome expression, the GEO databases. And I'm going to tell you that the sources of human stem cells that does have an effect on the co-regulation, actually, of the expression of critical telomere maintenance genes. And then just for fun, since it's the end of the day, I'll talk very briefly about hibernation and telomeres, just to end the day. And I'll talk about why we might be interested in that in the context of space. Basic science, a quick trip through. So telomere DNA is simple repeated DNA sequences cloaked with a whole lot of very well characterized and very studied proteins, just depicted in very schematic form here. And the repeated sequence is what nucleates this array of important proteins. And I'll introduce you more to the specifics of those in a few slides. But the idea is that you've got this telomeric DNA sequence and protective proteins. That is the shoelace tip, the protective aglet. Now, what's interesting is that this is actually a very dynamic structure, and there's a schematic sort of representation of this sheath as a kind of a bubble, but it's actually not a bad representation, because what we and others before us had learned is that there's very rapid movement of proteins on and off telomeres. They're very dynamic. So the telomeres you had a few minutes ago are not the same as the telomeres you've got right now in your cells, because there's been this rapid exchange of the proteins. And Zillmans looked several years ago at the exchange rates. We looked at it in our lab by Beth Samini, and what we found was that there's a really fast turnover half-lives of just several seconds of most of these critical telomere protective proteins. So your telomere is actually dynamic over time scales of seconds. So don't think of it as a little rock. Okay, so that's the proteins. And the other thing that's exciting and that makes sense of what I just said is that you can really see telomeres are liquid phase separation droplets. That's what the sheath is. Beautiful study out of Berkeley by Jack et al. A couple of years ago really showed that these are phase condensation, liquid condensates in the cell. Now, normally, telomeres are not stable for another reason, and that is that there is a normal process of loss of not just proteins turning over, but the telomeric DNA is much more malleable than typical chromosome internal DNA, and that's because it loses DNA from the ends through a variety of processes. First of all, thought of as just the end replication problem. You can't fully replicate the full ends of chromosomes. So that was thought to be applicable in replicating cells. But now there's much more understanding that there are other ways of telomeres being damaged, even in non-dividing cells, such as neuronal cells. And those damages are particularly exacerbated by oxidative stress, or reactive oxygen species that we've heard about earlier this afternoon. So even non-replicating cells are subject to damage. So the telomeric DNA is dynamic. This can happen over days or weeks. You can observe those timescales. But over the decades of human lifetime, there's also a general shortening of the telomeric DNA, and it's gradual, but it varies a lot, and that's the interesting thing, is the variability, and I'm going to talk a lot about that. Now, that variability has a real consequence, because if the telomere gets too short, as I'll show you, there are some severe consequences, very well studied for cells. And in fact, just to give it a little concreteness, Duncan Baird showed that it's about 3.8 kb is the minimum length of those repeat tract that, at least in a normal white blood cell, is supporting that whole stable telomere protective sheath. And as I said, normally, this is normal shortening that happens under normal physiological processes. But it's the variability that's really interesting, because if that shortening happens, a number of things can go on. Now, telomeres are dynamic. They don't just shrink. This would be bad news if they just did, but in fact, telomeres can grow. And telomerase, the ribonuclear protein, can elongate telomeres. So you'd think, well, the problem is solved. And what happens in humans is that while telomerase indeed can elongate telomeres, it's a very, very highly regulated both enzyme and system of things at the telomeres that I'll allude to in a moment. And so the action of telomerase is highly, highly regulated at all kinds of levels, and one of them includes transcriptional or gene expression levels, and that's what I'll end up, well, part of my talk will be about that level of regulation. And famously, telomerase is active in human stem cells, as it makes very good sense for their elongated potential to keep dividing, but also in cancers, particularly in human, advanced cancers. So it's striking that in many normal cell types, it's very, very tightly turned down as well, for various reasons that I won't really get into. So telomeres can get too short, and the consequences of them getting too short, that poor end now is not a lovely protected end. It's now an end that looks like a broken DNA, and the cell then responds to that with persistent DNA damage signaling. And it's not like an internal break in the chromosome that gets healed and then the signaling stopped. The signaling of DNA damage signaling, which has been well studied, has consequences, and I'll just outline them here. It causes cells to become senescent. That includes them not continuing to divide, but very importantly includes them also producing factors that can be pro-inflammatory. And it disrupts, of course, cell renewal capability, hence the ability to affect self-renewing tissues. But also cellular functions are affected, and many groups have contributed to this understanding, but for example, mitochondrial function is actually disrupted and leads to more reactive oxygen species, for example. So it can also lead to genomic instability as well, as those ends sometimes confuse with each other. So there's more than that, though. So the things that go on inside cells are bad enough when you have this signaling, but as I said, cells that are senescent, they've been well-studied profiles of what they secrete, and so they can have effects throughout the whole organism. And this can set up actually a vicious cycle. So you have the shortening, which I alluded to, you have this very short telomere, and it's sending a signal. The danger signal is, okay, I've got DNA damage. And the cells can have two effects. They're malfunctioning, but they also undergo senescence, in certain cases cell death, and that can disease tissue in different ways. But that also, because of these secreted factors, they can lead to inflammation, hence more reactive oxygen species, more damaging environment. So you can set up a vicious cycle here. And inflammation, as we know, also can have deleterious effects. So I'm giving a very sort of simplified overview of much more complex physiological processes. So, knowing all of this, does it matter, this gradual shortening that one observes overall in telomeres in humans? And human genetics, I think, has been so informative because it tells us that although this shortening occurs, it didn't tell you that it caused anything. But when you use human genetics, and again I'm going to summarize very high level, then you start to see real causality that you can hang your hat on. And so when you do GWAS analyses, you measure telomere lengths in lots and lots of people, hundreds of thousands. In our case, we did 100,000. We measured in cohorts, and you do GWAS, and a whole bunch of genes show up, okay, as being related. Their allele variants relate to telomere shortness, and actually the alleles, variants add up. And so, what are these proteins? Oh, by the way, we repeated some of these in a big cohort of 100,000 people. So, this is well established that if you have a variance of all these particular genes, they can have effects when you measure telomere length in humans, often done in peripheral blood mononuclear cells or leukocytes. Now, what are they all doing? Well, these are all players, and there are many more, that have well-established roles in telomere maintenance. And I've just depicted it, or this paper referred to here has depicted this. And so there's a very complicated set of things going on at telomeres. Up on the top is depicting, just in a very simplified form, the telomere structure. There are structural proteins. When that is deprotected, you get responses involving ATM, for example, DNA damage responses and other components. And you have effects there are known pathways of the biogenesis of the ribonuclear protein telomerase. That's coming up from the bottom left. And then there's the DNA replication, which has to have special things going on related to the ends of the DNA, as Tom's lab has been actually actively studying aspects of that. And so we have a whole lot of players. Here are the main ones for GWAS determining telomere shortness, if you will, in just normal populations of people, and we know what they're doing. So we can hang our hat on, yes, when we see these GWAS connections, they're actually making sense in terms of all of the known sorts of mini labs contributions to understanding what all these things are doing at telomeres, as depicted in this very simple set of things. Now, a lot of these are contradictory. They have to change partners. They have to do things at different times. And so it's a very complex dance going on at the ends of telomeres. So this whole turnover that I alluded to earlier of just the structural proteins kind of makes sense. This is very dynamic. And what it gives us is a really good list of important critical components to look at. Now, human genetics has another important use that's in the clinic, and there are sets of monogenetic diseases. In several of these are the green ovals, and it's over a dozen now, one at a time, any one of these when mutated in heterozygous form. So you have a good wild-type copy from one parent and a mutated copy in another parent. So even though you've got the wild-type copy, that nonetheless is not sufficient for telomere maintenance across the years of human life. And so what happens is that there's severe syndromes, severe diseases that can involve aplastic anemia, lung diseases such as pulmonary fibrosis, liver cirrhosis, osteoarthritis, and so forth. So there's a whole set of severe things, early onset kinds of things that happen. So these monogenetic diseases tell us that even 50% of your normal gene level of expression is not enough for human beings, unlike mice, which do fine with that. So when you get more telomere shortening than should be happening, as I said, you get this really bad consequence, the DNA damage response, and that's what's going on in these syndromes. So now, what about for the rest of the population who don't have these rare, luckily, but still very well studied mutations in these genes? So these just the common variants that make your telomeres shorter or not. So when you look at telomeres, you can say what kinds of consequences do you see of having shorter telomeres. We've already been instructed by the Mendelian are single gene mutations. They're very severe. What about in the rest of the population? And so this is, again, condensing and distilling a lot of studies where basically in the general population, the degree of telomere shortness, how much shorter your telomeres are than others in the cohort, is proportional to your risk of a whole set of diseases which characterize aging. And I've just listed several of them, and impaired immune function is certainly prominent among them, as is cardiovascular disease, but interestingly diabetes and dementias and osteoarthritis. So that's an association, doesn't cause anything, even though it anticipates incidence of these diseases. But human genetics using GWAS studies have been very informative, because now looking at GWAS studies, you can say that several of these, there's actually causality of the shorter telomeres. And the lung diseases include, for example, pulmonary fibrosis and also COPD now. And interestingly, dementias, Alzheimer's being prominent among them. That association has been seen. So we've got some causality there. Let's just talk for a moment about cancers, because cancers present a sort of interesting trade-off. And I'll just talk about the genetics, because that's the clearest with the cancer connection and telomeres. Now, in these haploinsufficient mutations, just half the amount of telomerase had this very severe effect of what I mentioned, these short telomere syndromes, but it also includes a propensity to a subset of human cancers. And these are just a subset of certain kinds of cancers. And what's really interesting is that Mary Armanius has shown that that impaired or that greater risk of cancers is actually because of impaired surveillance of your immune system. So your immune system is not controlling these incident cancers as it should be in this situation. So shorter telomeres are really bad, but for that very interesting reason. But also, if you have even just a little bit too much, say, telomerase expression or slightly more active production of some of those telomere proteins, making telomeres a little bit longer, some of the variant proteins, you get another set of risks for cancers. And these relate almost certainly to the ability of the cells with longer telomeres to have a longer runway to accumulate cancer-causing mutations. And this is a whole variety of different kinds of cancers. So there's a real trade-off in terms of cancers. So in terms of, well, where is this all leading us? So let's go back to thinking about what makes telomeres longer or shorter, better or worse maintained. And what's really important here, and the point I want to make, is that if you observe telomere lengths, I told you about genetic determinants of it, but that's actually a pretty minor part of the whole story. There are a lot of other factors that play in to observed telomere length, and a lot of them are events that happen in lives or society, influenced events. And I've just listed a whole lot of these here, and we got involved because of studies, and now there are multiple independent studies and cohorts, but we got involved with some of these with collaborations with my colleague Alyssa Eppel at the University of California, and Jolene in my lab has been really leading these collaborative studies, some of which we did with Alyssa, and many other groups have done the same thing. And so there's a whole lot of things that actually do affect telomere length. And because there's dose and length of duration of exposure to these things is proportionally affecting telomeres and they're getting shorter and shorter, we can say that there's causality is there. It's pretty hard to say it's the other way around. So it's both environment and genetics that are accounting for observed shorter telomeres or longer telomeres, whatever. Now, does that matter? And what's interesting is the study in the, this is a Scandinavian study of 64,000 people. And so I told you about cancers, and so you think, well, what's going to be better, longer, shorter? And in this study, what they did was they measured baseline telomere lengths just in white blood cells, and then they waited an average of 7 years and then said who died, and they divided people up into deciles of telomere length, so longer and shorter, shorter and shorter and shorter average telomere lengths. And then you look at the deaths, and you can see that they're really sort of dependent on the shortness of the telomeres. So this is shortness of telomeres is anticipating both all causes of deaths, and interestingly including cancers, as well as the other major cause of deaths, which is cardiovascular. So if you look, from down high up and look down, you can say, well, shorter telomeres are actually worse. And if you do multivariate analysis, these drop down somewhat, but the trends all stay in the same direction. So, shorter telomeres are not such great news for humans, even though we have the nuances of cancer. But overall, and perhaps it's because of this immune surveillance, average measured shorter telomeres are increasing the chances of death from all cancers combined. So that's just one of the many aspects of aging, and we've heard about the many aspects of aging throughout today, and the prediction power, as you saw from these statistics, is not all that strong, but it's real. But what if you combine telomere length measures with something else? And so I want to just give you a few examples of how this actually might be useful, because although telomere measures by themselves are not that predictive, they are getting more useful when you add them into something else. Let me give some examples. So there's a mitochondrial deletion that becomes more and more frequent in the genomes of mitochondria through a lot of tissues when humans age. And it's this particular, it's called 4977, just alluding to the particular region that's deleted in mitochondrial DNA. And you can classify people into high frequency and low frequency. You can classify them into higher telomere length tertile, lower tertile, and then you can just look at death. I'll just show you overall mortality, and what you can see is neither one has a really strong effect, but if you put them both together, you get a much more noticeable effect. It looks pretty additive. So telomere length is adding usefulness here, and similar result for actually cardiovascular deaths in the study. Here's another measure of aging, completely different one. You might think it's frailty. It measures, what is your strength and endurance and how fast can you walk and so forth. And even pre-frailty with just a couple of measures of losing your strength or endurance. If you look at that at baseline and then follow mortality, this is all-cause mortality, and also do the same thing with baseline telomere length and ask, well, if you have longer or shorter telomeres, what happens? And if you have long telomeres and you have no pre-frailty, that's the slower rate of mortality. The top curve, either one by itself, pretty comparable, and together additive. So you're adding in different aspects of aging, and you can see additive effects. So telomeres are adding something useful. Similar, this same sort of thing applies for full frailty and for cardiovascular disease. Now, what about a different kind of risk? And this is looking at dyslipidemia, looking at poor lipid profiles in blood of coal miners who are exposed to dangerous polycyclic aromatic hydrocarbons. And if you look at their risks, you can divide them into tertiles of how much exposure they had and what their risk of having a poor lipid profile is. And that's the front row is going from left to right, more and more exposure. And the front row is people who have short telomeres, and the back row are long telomeres, and the back row is people who have shorter telomeres. So in other words, if you have shorter telomeres, your risk is exacerbated. Now, this one is quite striking because this is a different sort of study, because these are not healthy individuals. These are people who came to MD Anderson. They came in, over 400 people in this study were followed, and at baseline their blood cell telomeres were measured, and also depression. And if you classified them into depression or not depression, and looked at either having short telomeres or depression at baseline, that's the top couple of curves underneath the very top curve, which is the no depression, longer telomeres, but together they were quite synergistic. So I think this is saying to me, telomere lengths by themselves, it's not much, but if you put it with something else, there's probably some really good opportunities to start combining it. And as cohorts get larger and databases get better, I think there'll be many more opportunities for this. Okay, so impacts are real. Now, those studies took years. We don't want to wait for years. So can we start to look at things that might be giving us an idea of trouble to come down the line by looking much sooner? And so now I want to tell you about how I've been looking at co-regulation of telomere maintenance genes. And what do I mean by these genes? I mean those core proteins and products that are involved in that complicated dance that's maintaining telomeres. And instead of looking just at the amounts of expression of their genes, which is what you typically do, you say how much expression was there, I actually looked at it in a somewhat different way, and not hard to do, and that was to look at the co-regulation of them across samples within different groups. So I'm going to just walk you through this because it's easy to do, and I'd kind of encourage this because I thought it's been quite interesting, and I'm going to take you through a number of vignettes. And basically, you're only going to have to look at patterns, pretty much. So the point being that what I'm going to show you, I hope might be useful for early warning of conditions that down the line will disrupt telomere length and maintenance and proper protection of telomeres, and hence the ability of cells to keep multiplying, and some experimental data that says these effects can be happening, quite quickly and quite directly too. Okay, so data mining of just, standard high-throughput RNA sequencing databases. I had to use bulk sequencing because single-cell sequencing has been challenging because these genes are expressed at very tight, low levels, even in stem cells. So the data are too sort of stochastic in single cells, and I'm hoping the technology will get better so we can do it with single cells. So these are studies where you're looking at cells of one type, but as a population, so bulk RNA sequencing. Okay, so mind you, here they all are, and there's about 40 crucial proteins, and they are different classes, and you've heard from about some of them. These are the telomere maintenance pathway genes. I'm calling them about 40 critical genes that I've showed you there, and a few more. Some of them are structural on the telomeres. Some of them are to do with the DNA damage response, the proper response to telomeres that can sometimes prompt telomerase to act on telomeres or protective proteins to act differently. The telomerase structural, the ribonuclear protein structural proteins, they're biogenesis genes, and TERA, which you heard about this morning, those RNA transcripts that go through, what regulates those transcripts that go through telomeres. So let me just give you a few examples. So this is real life, this is UCSF, a big study. These are fairly healthy people, and each spot is the peripheral blood mononucleosides of one person. And what I'm plotting is, across this whole set of samples, is the level of expression of two of these telomere proteins. They're called, the names really don't matter, but the ACD and TNF2, they're crucial telomere shelter-in proteins, well characterized, they protect the telomeres. And what you can see is that they're expressed proportional to each other. Now, let's think about this. These are healthy samples, but there's always inherent variability. But what this looks like is there's a sort of a compensatory nature to this, that if it's a little bit lower in one, it'll be a little bit lower in another. The converse is true for another pair, ACD and POT1. POT1 discovered initially in Tom's lab as a fission yeast protein, protector of telomeres. And that's inversely expressed, right? And there's another pair of proteins shown on the right. This is POT1 and another one called TNF1. So TNF2. So basically, pairwise, you can see different relationships in this cohort. So now, let's see if we can extract some interesting things from this. So this is a study out of Stanford, and they're looking at about 100 donors, and they're looking at multiple cell lines that they've made from normal T cells. They've activated T cells, they've made induced pluripotent stem cell lines, and then they passage them, and then they do RNA-seq on them. So I extracted from their databases these 40 genes or so, plus some proliferation genes. Now, if you look at a pair of these genes, just as I showed you, you can see there's a sort of scatter, right? There's not a whole lot going on. But let's divide them up. And in this paper, what they did was they said, what does insulin resistance do to the ability to make induced pluripotent stem cell lines from these individuals compared with insulin-sensitive people in this cohort, and age and sex, and what influences might there be that impact? They were interested in stem cells, but what influences might there be, I was interested in, on these telomere pairwise correlations. So let's just take an example just to simplify it. So we've got about over 300 samples, multiple samples, and they've all been, cultured in vitro, RNA-seq is done. So let's look at, the males, let's look at the younger ones, they're in about their 30s, and the insulin-sensitive and the insulin-resistant. And now when you look at in these subgroups, now they actually have different relationships, okay? So that's a pairwise relationship between two of the telomere expression genes. It's already different, and I'll show you much more. So what I also like to do is look at the genes that are related to proliferation rate, because that's a good, telomeres have to be replicated, and so they should coordinate with cell proliferation rate genes. And there's a whole bunch of those known too. Here's one example, MCM6. And again, in these two subsets, the relationship of just a pair of these telomere genes is different from, or one of these telomere genes is different in the insulin-resistant versus the insulin-sensitive group. Okay, so now let's extend this to all these genes. So lots and lots of genes down the left column. You don't have to look at individual things here. Sit back, enjoy. Just I have these 40 or so genes. There's 30 or so proliferation genes that I show in some of these. And then each column is one individual's RNA expression levels for that gene. And so those are lots of individuals. And I'm always going to divide them up into two groups. There's going to be all of the individuals who fall into the healthy, favorable group, and the ones in the more adverse group. So it's always going to be the healthy ones on the left, and the adverse ones pretty much on the right. So now let's look across it all, and now we'll look at all the pairwise combinations, right? So we'll get a big 2D heat map of all the pairwise combinations, and then I cluster them, and then I say, is it different? So we're looking at differences in the co-regulation of these genes, and we know that their transcription levels are critical. We've learned that from the human genetic studies. So let's look at the source of the stem cells. In this case is people who are insulin sensitive versus insulin resistant. Data mining, as I said. And so look at the patterns. There'll be telomere genes up the top, proliferation genes down the bottom, and then the matrix is, across the top is the same genes, and then we just look at the 2D heat map. And this is, you can see these very nice clusters. You can see telomere genes. There are telomere genes that co-regulate. That's blue. Telomere genes that anti-co-regulate with each other. That's red. And a whole bunch of those co-regulate with proliferation rate genes, but some anti-proliferate, right? So there's a pattern. So that's in the healthy individuals. And in the same culture conditions generating the induced pluripotent stem cells, everything from the same matched group, but now insulin-resistant people, chaos. So that was really quite intriguing because, there it is, right? They aren't regulated in the same way. So now, this is aging. So now, insulin sensitive, and there's other data, but I'll just show you this. Now people in their 60s, and you see it's sort of become a bit more chaotic. I mean, it's not overly lost, but aging has certainly lost some of that regulation. Okay, so now we're off to the races, and so we say, well, okay, insulin-resistant cells have, been living a long life, months, years. They've probably got a lot of epigenetic changes and so on. But we know that in insulin-resistant cells and in diabetic cells, they're exposed to different levels of glucose, for example, because physiologically the glucose will be higher in people who have metabolic syndrome or diabetes. So can we look directly at that? So now I'm going to just take you through some experiments where people have been looking directly at certain conditions in culture of cultured cells, so they're well matched with an adverse condition and a healthy condition, low glucose or high glucose being the healthy, the low, the high is the unhealthy condition, or stress hormone. And we know stress, we know insulin resistance and shorter telomeres, really go together. We know that increased stress, dosage and chronicity goes with causes shorter telomeres. I want to show you one drug, a common headache drug, which was quite surprising. So I'll just take you through these, and these are all direct, exposures in vitro. So let's have a look. primary hepatic cells. They're put in vitro, and then you expose them to either the healthy glucose or just to physiological but high unhealthy glucose levels. Look at the expression of the genes, first of all. And if you looked at gene expression profiles and said, is there a difference, you'd be very bored, because that line across is 1, and that's the ratio. So you'd say, well, nothing's much is happening. So you'd say, okay, I'm going home. But don't go home, because look at the co-regulation in the healthy case, and you've lost it. In the just 4 hours of glucose is the difference. This is just telomere genes I'm showing here too. So a very relevant cell type, pancreatic human islet cells. These are primary cells, normally cells, short-term culture. Just treat them with the 2 different concentrations of glucose in vitro. Big difference in the coordination among the genes. So another type of perhaps relevant cell type is human-induced pluripotent stem cell-derived glutaminergic neurons. So this is another study I pull out, and I look at the effect just of 24 hours of looking at a stress hormone. And you can see that the pattern has become somewhat disrupted even just with this 24-hour exposure. Another interesting one is, it was an intriguing study. They were looking at paracetamol. That's the European name for acetaminophen, which is Tylenol, right? And just a twofold difference in the concentration of exposing these cells to just a few days of this, seemingly harmless drug. You actually see that there's a disruption of the telomere co-regulation here. So this is quite a good way of sort of catching differences in co-regulation of these critical pathways. We know there's a lot of inputs into all the sort of transcription factors and signaling that's going in, but we can see it in this case. And in each case, well, not yet for the paracetamol, but we know that they all correlate, the adverse one correlates with shorter telomeres and disease risks. And this was an interesting study with just fibroblasts, primary differentiated, of course, fibroblasts. And what they've been able to show that, in this paper whose database is referenced here is that, in fact, if you have softer matrix, you don't get such good proliferation. But the proliferative lifespan, even though the proliferation is slowed down, actually the lifespan is sort of slowed down. And so you reach senescence quicker, replicative senescence. Telomeres run down in these cells because they lack telomerase activity. So what about the same analysis in several samples of the cells early and late in passage exposed to stiff and to loose matrix, and the stiff matrix is the more favorable condition. And you can see shifts even at the beginning of the early passages. You can see in this correlation patterns, you can see, just look at the patterns of where the reds are, and you can see that there are shifts already that are happening. So this is quite striking because at this very early passage stage, you wouldn't know anything was going wrong. Okay, so interesting ideas for regeneration. For example, if you expose myoblasts, I'm just giving you vignettes in vitro to aged versus young serum, the telomere pattern in the young serum is somewhat different from what you see in the older serum. It's a confusing pattern, but you can see the patterns have changed already. Okay, so perhaps this kind of way of just simply looking at the patterns of gene expression in multiple samples is useful for early warning signs. Things aren't going well. So I just want to talk about another fascinating study that came from Svante Pääbo's lab and collaborators. And in this, what they're looking at is the resilience of cells to insults, to toxins, particularly ones that are caused by things that are caused by smokiness from cooking fires. And so people have evolved an aryl hydrocarbon receptor gene, which is in all modern humans different from that in Neanderthals and Denisovans. And it's an alanine to valine change, and it makes the active site less able to bind the toxic ligands. And so what they did was they used human embryonic stem cells and engineered them by CRISPR to have the ancient form, which we can't find on the planet Earth right now. But they asked what happens, and they found gene expression changes. But I looked at their data in this same way and said, what happens to the telomere maintenance? So here's the past, and maybe this will be an interesting thing to think about, what we might think about as we look at future challenges, because the idea is that this has evolved based on a on a change in humans' exposures to smokiness. This is all the gene expression data, just to tell you nothing changed in terms of levels of gene expression. So here we have the modern day, just no challenge, just the control. Here's the correlation pattern. And here we have it where they've taken away they've re-ancestralized they've replaced the modern gene with the ancient gene. So they've lost, they've really changed. And, this is the inhibitor of the aryl hydrocarbon receptor. So that's the pattern you see. It's really just changes, just by changing that, you can change the correlation patterns, but it looks fairly like what you get with just the control ancestral gene. So in other words, the ancestral gene looks as though you've inhibited the aryl hydrocarbon receptor. So, okay, that's interesting. So it converts it into the ancient form in terms of telomere maintenance. But what's fascinating is that when you subject modern cells to a nasty toxin, this benzyl pyrene compound, which causes all sorts of damage in cells. This is what you get. So this is the normal modern response. And now we look at the ancient response, and you get this tremendous, turn on of all sorts of genes all at once. So in other words, there's a terrific response. They're super sensitive to this damaging environmental toxin in the ancient form, but the modern form is much more resilient. Okay, so I'm just going to finish up with a fun thing. We talked about microgravity and space travel. Okay, now when you think about going to Mars, and Ray Ban-Nawi referred to this, it takes months and months, and so people are really seriously thinking about both microgravity effects and also hibernation. So I just want to show you one effect of microgravity. This is now a simulated microgravity on an experimental setup on the planet Earth. Again, you can see that the telomere coordination is changed, whereas in the bottom right-hand corner of each of those, that's the proliferation rates, and they stay, very nicely correlated. But the relationship of the telomere genes to them and to each other, as you can see, is changed just by exposure for a few days to this simulated microgravity. This is immature dendritic cells, really critical immune system cells. And I'll finish up with a very fun sort of futuristic thing. Imagine you're going to Mars. It's very seriously contemplated by nature. By NASA, that one would have to go into hibernation to preserve resources. So what can we learn about hibernation and telomeres from the closest relatives on the planet who are primates? And there's a very few primates that actually hibernate, but in Madagascar, there are lemur species that hibernate in warm weather because there's a hot, dry season, they can't get food. And so Duke University has a whole lemur center, and Dana Smith in my lab collaborated with them, and we looked at telomeres in the whole time sequence, the longitudinal sequence of several lemurs, where you looked at the beginning of hibernation, middle of hibernation, as they ended hibernation, and then they come out of this very slow metabolic state with torpor, and they wake up, and the metabolic rate goes up, and so we looked at 2 further time points. And this is what happened to the telomeres. They do really well in hibernation, okay? So that's good news, but they crash back when they come out of hibernation and have this huge metabolic burst. So I think we should think about these kinds of things for the long-term health of people who are undergoing hibernation. So on that futuristic note, let me just put the summary slide up and tell you that I think we can look at telomere maintenance. It might be very useful for early warning of things that we know will be very likely to result from dysregulation and lead to problems with long-term maintenance down the line. So thank you for your attention at the end of this day, and I'm very happy to discuss over, drinks, wine, whatever, tomorrow, any of this at all. Thank you.